Wonderful presentation. And now we move to the scientific part. It's really to the keynote lectures. Oops. It is my true pleasure to introduce Professor Samuel Stupp from Northwestern University. Luckily, we are short in time. Otherwise, I would need about half an hour to introduce you. But I would say that I cannot think about a better person to represent the interdisciplinary ideas of chemistry and biology and engineering and energy research than Sam. For me personally, he has been a role model in the application of so many interdisciplinary fields. He has been recognized by many awards, many membership in academies. Again, it's you can see it in the booklet. It's not in the booklet, but he's been recognized as one of the highest cited chemists by the ISI, and many more recognitions. Sam, it's really a pleasure to have you here. Thank you.
Now? Perfect. Okay, so I hope that you heard that I was giving my compliments to everybody here in the front row for the great achievements of Tel Aviv University. Okay, so the lecture this morning is about soft materials and their impact on the environment and their impact on aging as well. So the research that I do in soft materials connects to the idea of self-assembly. So these are molecules that could exist in the atmosphere or in liquids or even in solids, and they are programmed to create structures, very well defined, and those structures have a function, which is hopefully of great value. And this is actually a biomimetic idea because this is the way that nature develops all its functional structures. And the first collision we had with this field actually goes back to 1997 when we discovered molecules that would self-assemble into this mushroom-shaped nanostructure, which then would proceed to stack in the way that would be considered absolutely irrational by a mushroom farmer trying to pack mushrooms in a box. So the packing arrangement was stem to cap, stem to cap, stem to cap, and this is called a polar arrangement, and therefore the surfaces of these films were completely different chemically on the top and the bottom of the film, regardless of the thickness of the film. So we could make things that could repel water, for example, on the top, and then become very water-loving on the underneath surface. This was all produced by self-assembly. And so why should we program molecules for self-assembly? And so the reason to do it is to access functions that are very special, for example, in hierarchical structures, which are all over nature. And it's not possible to create them using traditional chemical methodologies like polymerizing molecules or mixing components. We want to achieve great functions, therefore, using self-assembly because, for example, if you think about this field of regenerative medicine, which connects to aging and is one area that we have been very interested in, or novel therapies for disease, you need unique structures with unique functions. And it is also possible, for example, if we are in this quest of using the sun as a source of energy, which is very important for sustainability of the planet, it is important, again, to have unique structures. And this is all why self-assembly is an interesting opportunity, because it opens up many new possibilities. The other third reason, which is very important, which is something we have not yet been able to achieve in chemistry and in material science, is to create materials that emulate living matter. So they're synthetic, but they emulate living matter. So what are these possibilities? For example, hierarchical structures, very quickly. I will show you what that means very briefly, using the example of bone, using the example of bamboo. In plants and in animals, you have many levels of structure that are connected to each other. So this is what we call hierarchical structures. And it is one of the things that self-assembly tries to achieve. Now, materials that emulate living matter, which is a big part of these unique structures, what does that mean? Well, it means, for example, achieving materials that exhibit motion. They move as though they were alive, and they do so across scales. So not just at the level of molecules, but at the macroscopic level. Regeneration is a property of living matter. You know, you have a wound on your skin, and the skin will repair. You fall, a bone breaks, the bone will repair. So we don't really have yet systems that do regeneration, synthetic systems that do regeneration very efficiently, but we are making great advances. So healing capacity. The conversion of
It will work in a second to have enough wire.
Okay? All right. So I was saying that the conversion of photons to food, which is an amazing capability created in nature, is something that we connect with living matter, and it is one of the objectives that we have in creating these materials. Replication, morphogenesis, which means the change of shape, and also programming materials to be biodegradable and reusable when they reach non-functional states, which means death in nature. In nature, plants and animals die, and all the components are recycled again. So how can chemistry and material science, which are my two fields, how can we help? And the answer is to create molecules that are programmed to not just self-assemble into a structure, but to do so reversibly is very important. And this connects to the field of supramolecular self-assembly, and also it requires strategies to have control dynamics in synthetic systems. Okay, and why is the field important to science and to society? Well, for those of you in science in the audience, you will appreciate that this helps us understand systems that are far from equilibrium. This is a very important scientific objective. But on a more translational side, delivering the molecules and the materials that we need for healthy aging. This is really critical moving forward. Since people now live nearly a century, you need to achieve healthy aging and also face the energy challenges and protect our planet. So I hope that's enough motivation to now begin to tell you a little bit about the science that we do more specifically. And so this is a list of areas, which of course I don't have time to talk about all of them, but it's a list of areas that my lab at Northwestern is working on in the space of soft materials that emulate functions of living matter. So the strategy that we use is called in chemistry and in material science supramolecular chemistry or supramolecular material science. So supramolecular systems means that you design a molecule to interact with its neighbors in very specific ways. And this is accomplished by things like hydrogen bonds, electron overlaps, electrostatics, steric forces when molecules repel each other. And the reason this is interesting is because these bonds are weak bonds. They're not strong bonds. And because they are weak bonds, that means they have a finite bond lifetime. And by having a finite bond lifetime is when you then achieve what we would call dynamic soft matter, things that move, things that reconfigure, that adapt, and they respond. And the most important types of substances in this space are the so-called supramolecular polymers, which means that the molecules create a polymerized structure, but the components are not bonded by strong bonds that are not breakable, basically very, very strong covalent bonds, but weaker bonds. Okay. So I'm going to give you now some examples that are perhaps more specific. One of the things that we wanted to do in the space of converting photons of the sun to food was to imitate the materials that are involved in plant photosynthesis, which means that you have to have the capacity to absorb light in a specific way and to transport charges as a result of that light absorption, and then somehow use those charges to do chemistry so that you could produce food, you produce something useful. And so the objective that we had here was to create materials that makes us think about leaves, for example. Leaves are, in trees, are highly hydrated materials. They are hydrogels in a material science sense. And so we wanted to create materials that, like leaves, have the capacity to absorb visible photons from the sun, and then they could do something useful. So here is a photograph of the first material that we developed to do this. This is a red material. The fact that it's red already tells you that it's going to absorb a lot of light from the sun. And the model that we used was to, well, it starts with a chemical design. This is the molecule that we developed. This red part is what makes the material absorb light from the sun. At the nanoscale, these molecules make these ribbons that you see here on the slide. And these ribbons are water soluble. And you see them here imaged by cryotransmission electron microscopy, which means we just sort of freeze a solution in a water solution, and then we can image what they look like. And so they are ribbons. Now, one of the things, and this is now going to be my connection to the Dead Sea, what you're going to see in the next slide. This molecule makes these ribbons. They absorb light, so that they are going to absorb light in a very special way. But we discovered that when we add highly concentrated salt, so this is the Dead Sea connection, you add salt, and so you watch now what happens. So these are those ribbons that absorb light from the sun in water. This is in water. It looks like a Cabernet Sauvignon wine. You add 3 molar sodium chloride, and this immediately converts the material to a soft solid. So let's see if we can get this activated. These are my graduate students talking, actually. Let's see if I can. Yeah, there you go. You add the salt, and this converts it into a soft solid. So this is going to be a hydrogel, like a leaf-like material, which will then have some interesting function. And in fact, when you add the salt and you go to this to the synchrotron, which is this huge advanced photon source in Argonne National Laboratory, you look with X-rays through this hydrogel, and when you add the salt, we discovered that these peaks appear, which are called wide-angle X-ray peaks, which indicate that there is a crystallization of the molecules in those ribbons. That crystallization of the ribbons was found by us over the last few years to be key in being able to use this material to produce hydrogen fuel. Okay, and so, and that's because the so-called excitons that are necessary for this process move very rapidly and efficiently when the crystallization of these molecules takes place. So you see here a cartoon. So you are inside the material. So it's blue here because it's all water. This material is largely water. But you see here the ribbon, which is this white structure, and these yellow molecules that you see here are catalysts, which are going to be able to convert protons, which come from vitamin C floating in the water, and it's converted to hydrogen fuel. And the hydrogen just bubbles out of the system. You see here the experiment in our lab looks something like this. And this is a measurement here in this graph of the amount of hydrogen that you are producing as a function of irradiation time. So you're irradiating with visible light, and as you do that, hydrogen fuel is being produced inside the material, and it's bubbling out of this soft hydrogel. Now, because this is a solid material, then we are able to, I'm not sure, the flowers might be on the way here, but you might be able to see here that this material can be formed in very specific ways because it is a soft gel, and so we can use 3D printing, which is a modern processing methodology, and create these structures to optimize the conversion of fuel. Now, a very similar, okay. All right. A very similar concept using.
This is Israel. You have the mayor
Okay.
doing this.
The mayor doing this. Okay. How's that? I actually sent Mira a text. You just saw it? I sent her a text from my chair there. I said, maybe they can move the flowers. Okay. Thank you. Yes. Now you have the proof. Okay. All right. So just to show you a similar material, this time is a yellow one, was engineered recently in our lab not to produce hydrogen fuel, but to take carbon dioxide from the atmosphere and then convert it either to carbon monoxide or to methane. So we are able to make, now carbon monoxide, of course, is a poison, but carbon monoxide, which can be made from CO2, can then be used to make useful chemicals. So you could use it for chemical production, and or you could, and of course methane can also be a fuel. So what's interesting here is that you are taking a pollutant from the air and using the material to convert it into something useful like a fuel. Okay. Or to make interesting molecules. Okay, now a change of gears. Let me show you some examples that connect to regenerative medicine. All of you in this room should be interested in this, so pay attention now. Now it's not CO2 or hydrogen fuel, but it's you. Okay? So regenerative medicine is the medicine of the 21st century because you have people living 100 years. We all want to live 100 years, and we want to live them well. Okay? So we're going to have many things to achieve that, but one of the things we need is the ability to regenerate key tissues of our bodies. Okay? And this is both not only important for a happy life, a healthy life, but also it's an economic issue because, how can you keep humans on the planet living 100 years in a dysfunctional state? That's pretty tough. The economy will not be able to accommodate this, right? Okay, so we are interested in all of these things. We're interested in the brain. We're interested in musculoskeletal regeneration, which means making new cartilage for bad joints. We are interested also in organs, for example, kidney regeneration. So we work on all of these areas in my lab. And so the way that we do it, again, is to use the concept of self-assembly that I introduced at the beginning of the lecture, but create forms of the materials that are highly bioactive, which means that they can signal cells in order to regenerate tissues. And so here you see where the inspiration came from. I said that the self-assembly is a biomimetic strategy in chemistry and in material science. Well, this is a cell. This is kind of a Picasso representation of a cell. And here is its membrane. And this is the interior of the cell. This is the outside of the cell. This is called the extracellular membrane, the extracellular matrix. The extracellular matrix contains a lot of filaments, very, very thin structures. So we learn to create these very thin filaments to imitate the ones outside of the cell using self-assembly, using molecules that could be programmed. Here you see an electron micrograph of our fibers. So these look like components that you are likely to find outside of a cell, but these, of course, are artificial, and they are made by self-assembly, by supramolecular interactions. They are supramolecular polymers. You see them here. And so what happens is we have this molecule that you see here is called a peptide amphiphile. And we develop these molecules to self-assemble into a filament. So you see them here. The black space is water. So you put these molecules in water, and they self-assemble into a very thin filament that measures only 10 nanometers in diameter. Over here, you see how the self-assembly process occurs. This purple segment that you see here in the structure is where the instructions are based on amino acids, the instructions to create a highly elongated filament. Now, I'm going to show you. So these filaments, we found, after several years of research, that cells really like them. And particularly, if you put a signal right here, a biological signal, which is displayed on the surface, you can basically activate cells to do things that they don't do naturally. So for example, you could cartilage, that wonderful material that doesn't grow anymore after you turn 18. So it's all over. After 18, forget it. Cartilage will not grow again. So we can get cells to produce cartilage, the specific types of cells, by putting the right signal right here on this molecule and then having self-assembly occur this way. But the interesting thing is that unless they are an extremely long, thin filament, it doesn't work. So that means that cells, from an evolutionary point of view, are I guess, wired to recognize tiny nanoscale filaments in the extracellular space. And this is part of the signaling. So I just want to show you, in the next slide, there's a picture I took yesterday when I was walking with my wife, Deborah, on the water. I took this picture right here because it really connects to these filaments that I'm talking about. If you place these molecules that we invented in the Dead Sea, they will become infinite fibers, infinite. Because, I mean, there is the reason is, for those of you in physical sciences in the audience, the reason this happens is because we, by design, put charges on these filaments so that they can be soluble in water. You need them soluble in water. If you're going to inject them into an individual, they have to be soluble in water, not in an organic solvent. So they have charges. But if you put them in the Dead Sea or you add salt to the water, then you screen the charges. And in this way, by very, very strong screening of the charges, now bonds form among the molecules like hydrogen bonds, and they become infinite. So these are, thermodynamically speaking, infinite filaments, but provided they are in a salty environment. And so in the Dead Sea, they will be infinite filaments. Okay. And then the way they signal cells is shown here. You see here one of the filaments. And so we put specific signals in yellow right here. And these signals talk to the receptors. So these are proteins on the surface of the cell. And these are being signaled, and then great things will happen. And the materials themselves, they are now soluble in water. You see them here. So what we're going to do is do the equivalent of putting salt, going to the Dead Sea. And watch what happens when you add the salt or when you decrease the charges. The liquids then become, as you see here, it's water, but then they become a gel, bottle upside down, and I can reverse this again. So they go back and forth between being gel, and so when they're in a salty environment, they gel. So the concept here was that if we would inject this into an individual, our fluids are like the Dead Sea almost. Very salty. We're very salty people, salty animals. And if you inject this into a patient, then the material, which is liquid, would immediately gel and then it would be localized in the right place to do what you want the material to do biologically. Okay, so what have we done with these materials? Well, in this paper back in 2004, that was our first success with these materials. This cluster here with blue dots are neural stem cells. They came from embryonic mice, from the brains of embryonic mice. And we discovered that when our filaments touch these neural stem cells, they make new neurons. And we have perfected this now. We've been sending these materials all over the world. People do experiments with them. And they love the fact that they can make neurons so easily without using any proteins. Just the signals that we put here on the surface of the filament will convert these cells into neurons. These green extensions that you see here is a signature that they are new neurons. Another thing, and I should say that, of course, this is important because the way we're going to approach Alzheimer's and Parkinson's and many other neurodegenerative diseases is going to be making new neurons. And those new neurons, hopefully, will come from individuals, from the patient. They will be converted. Let's say you'll have a skin biopsy, convert those to stem cells, which was the subject of the Nobel Prize 4 years ago by Yamanaka in Japan. But then we'll need our filaments to convert the stem cells to become neurons. So they will be Mira neurons or Yossi neurons. in a specific way. Yeah. Okay. Another thing that we do is signaling for bone regeneration. So, and we are targeting specifically spinal fusion. So here's the molecule that does that. A lot of chemistry here. I mean, we perfected this pretty well over the years. And so what you see here is the spine of a rabbit, okay? And you see this blue region. This blue region is new bone, okay, in the spine. Now, why is this important? Well, because talking about aging and healthy aging, 80% of people in the world have back problems and disc degenerative disease. And the regenerating the disc is a subject probably for the 22nd century maybe, because it's very, very hard. But what we can do is fuse the vertebrae. And so when the disc doesn't work anymore, so then that avoids the pain, you gain mobility, you become a normal person again, you can walk, you can enjoy life. So we've done this using a material, a specific material. And the reason I'm very excited about this is because this material is now in the process of being commercialized by one of the largest companies in the world in the biomedical space. And so I'm very proud of that. And this particular material works with this protein called BMP2. It's a protein that makes bone, but it's been used already clinically, but it has many, many dangerous side effects. What our material did was to reduce 100 times the amount of protein that you needed to get spinal fusion. And so we're very excited about that. This is like, so it works in the pig, in the rabbit, and the rat. Actually, the rabbit is the most predictive one of what's going to happen in humans. So that's another example. Okay, and I just want to show you what the material looks like. So you go down here. Now you can see without the flowers at the bottom. And then you see let's see if this works. You see? It looks like toothpaste. You squirt it out of a syringe. It's like a toothpaste. You put it between the two posterior lateral processes of the spine. And 12 weeks later, there will be beautiful, healthy bone bridging the two vertebrae. So that's how it works. We have another one, this one right here, which behaves as though it were brain-derived neurotrophic factor. All the neurons of your brain are addicted to this protein. And if you could have this protein as a therapy for regenerative processes, it'd be fantastic, but you cannot use the protein because the protein disappears very quickly. So we developed a filament, which you see here, that actually talks to the receptor of a neuron, and the neuron thinks that it's touching the protein, but it's not. It's touching this filament. And this also connects to the problem, one of the problems that we are very passionate about, which is spinal cord injury. So the spinal cord is also part of the central nervous system, just like the brain. And so I've been very interested in this particular problem for quite some time, to be able to reverse paralysis or to prevent paralysis after a traumatic accident, for example, right? So, I mean, curing spinal cord injury or avoiding paralysis in individuals is a major part of healthy aging and be able to avoid disability, very serious disability. So now I'm going to show you, this is a very interesting new thing that we have. So this is the filament that you saw before, but watch what I'm going to do with this movie here. You see the movement of the molecules here? We have recently discovered that by actually designing the speed with which these molecules move, that can make the difference, with the proper signals in the recovery from spinal cord injury, which is really an interesting finding. You see that here in a graph. So this is a graph that shows the progress in functional recovery after spinal cord injury as a function of time. So what we do in the lab is, if you come to my lab, then you see it. We have a room where we use rats, and we induce an injury in the spinal cord, or a mouse. I mean, we do both mouse and rat. And you see here, this is the phenotype, or this is the animal that has had the injury. So what you do here is you see, let's see, okay, there. Okay, so you see the hind limbs are paralyzed. So the animal just drags the hind limbs. That's the injured animal. And in fact, if you follow the recovery of this animal, is described by this red curve. In red and in blue is the exposure at the site of injury of our materials, moving very slowly or moving very rapidly. This is something that a few years ago we would have never predicted. Okay, and so you see here the phenotype when the animal doesn't move very quickly. When the filaments, the molecules don't move very quickly, there is some recovery, but there's not very much. And then when I go to the blue, is when the molecules are moving very quickly. Now you see the recovery so the animal is now able to move the hind limbs and is able to walk, which was unable to in this case. So this was done just by basically injecting in water these supramolecular filaments and then watching progress over time. Okay, so now how am I doing on time? I guess I should finish up. Okay, so I'm gonna just close the lecture just by showing you maybe just a very, I'll show you this slide right here because it's the subject of a very recent paper that we published in Science magazine about 3 months ago. And I figure there might be some computer scientists in the audience here, right? And so I just want to show you a new type of material that we developed. And this is a computer simulation that predicted exactly what we saw in the experiment. And that's the subject of this paper. And so you see here a bunch of filaments, maybe not so different from the ones you've seen before. But in this simulation, which took about 3 to 4 weeks using 8 processors, describes what happens in about a millisecond. So this is a very, very expensive simulation. And you watch this movie, and you see what happens in time is the molecules form a hierarchical structure. That's how when I opened my lecture, I talked about the hierarchical structures. Here with high resolution, you see them better. And so in a millisecond, the molecules are, many molecules are arranging themselves into this hierarchical structure, which looks something, let me move this way, looks something like this. So this is a hierarchical structure, is a bundle of many filaments. And what's really interesting about this is that this is a reversible process. Okay, so we can, and this was unprecedented, of being able to build a hierarchical structure that is totally reversible by, for example, adding molecules to the material. Okay, so before Uri gets too nervous, I will advance here and just show you, finish up with just something fun to look at, which is how we are building on these platforms of self-assembly, integration of supramolecular structures with covalent polymers to create robotic materials. That is, materials that emulate living matter by exhibiting autonomous motion. So this red material that you see here, when you shine light on it, you can see what happens. It just bends like that. We have another one that walks. So also with the application of light, this material is walking, light, dark, and it advances, and it's a macroscopic structure. This one, which is the one I'm really excited about, and this is actually real time, so this is not an accelerated movie, this is real time. This is a structure that has this shape, it's in a tank of water, and it is in this shape because of the light, and then we are applying magnetic fields on the outside. So this is all designed, very carefully, molecule by molecule, and you can see this material walks. It has autonomous motion as a result of exposure to light, visible light, light from the sun, and magnetic fields. So we're very excited about that. And so I'm just going to close my lecture by showing you 2 pictures. I like to joke sometimes, this is my group, but it's not true. It's really not true. But it was a celebration. It was a celebration, a couple of years ago of my lab. And the point I want to make here is to do this type of research, convergent research, which involves many fields. Of course, you need the students and the postdocs. They're critical. And you'll see next their picture. But you also need your colleagues, Your colleagues are a part of that process, And so I guess there is one Israeli here in this picture. You might recognize her. She's next to me, right there. Who is that? Can you recognize her? That's Leah Dadi. Leah Dadi right here. And there's George Whitesides, which has always been a very close friend and great source of inspiration. And there's my wife, Deborah, right here. So she's in my group also. And anyway, but these are my students and my postdocs. They do everything that I talk about. And I'm very lucky to work with a group of extremely talented, creative young people, and which is a fantastic privilege to have. Thank you very much for your attention.
Thank you very much for this truly wonderful talk. Yes, truly wonderful talk. Questions, please. Yes.
So I have a question, just a curiosity question about hierarchical structures.
Yes.
How does that jive with our understanding of entropy? I mean, they're assembling into ordered arrays, but entropy tells us everything should go in the opposite direction.
Well, actually, some of the structures, even when they are hierarchical, strangely enough, they recover entropy. Okay, which is a very simple example that I can give you is when you have molecules that are elongated and rigid, and they all align in one direction. The fact, and this is oftentimes the first step in creating a hierarchical structure, allows the molecules to move, to translate more easily. And so there is actually greater entropy in achieving that order. And there are many other examples, like that. So, but of course, hierarchical structures are not just about entropy. I mean, hierarchical structures are about enthalpy, right? So you need favorable interactions. You need to condense molecules and lose enthalpy. But there's always, there are many surprises about the role of entropy, actually, in creation of hierarchical structures.
Thank you. Sorry for letting you run. I was interested in your, at some point you showed some pins on your filaments that say this thing signal. So what are the things that signal in these filaments?
Okay. What signal in these filaments are typically sequences of amino acids. Okay, so we design sequences of amino acids that have the capacity to activate a receptor. Okay, so that's the most common type of signal. Another type of signal, so basically you have like a short peptide that's attached to the filament, and it interacts with a receptor, which is a full protein on the cell membrane, and that contact triggers a signaling cascade in the cell that ends in the expression of genes. Yeah, so basically we create protein mimics. In another form of signaling, what we do is we create a small sequence of peptides, of amino acids. We create a peptide that recognizes a specific protein, and then we let that protein signal the receptor. So those are two very common ones.
One last question, because we are really very short.
You're not going to make me walk up all the steps. Thank you for your talk. Maybe because I'm a biologist, I'm going to ask an environmental question.
Okay.
And that's about the first part of your talk.
Okay.
When you converted the CO2 into methane and you formed a gel,
Yes.
is that gel, what would that do to the environment? I mean, are we forming energy and then hurting the environment with gels that won't break up?
Right. Okay. So those gels, in an ideal world, right? And we are trying to go in that direction, but this will take a little bit of time. The idea could be that the gel is created with biomolecular structures. For example, sugars, or could be amino acids. It could be nucleic acids. I mean, you could create them that way, and we have already shown that we can do that. In the original work that you saw on the lecture, those molecules are not made with biomolecular structures. But remember, you also need the chromophore, because this is what's going to absorb the photons from the sun. So I think the key is to make them from small molecules so that you can recycle them and they can break down. So these would not be things that would create plastic pollution or anything like that. These are hydrogels made of small molecules that can be easily broken down and recycled.
Thank you very much. It was wonderful.